So folks, welcome. Let's hear just the announcements. Hope you have a really good, safe, warm Thanksgiving break. And folks, what we'll do today is we'll finish the first PowerPoint on virulence factors, and we'll be focusing on bacterial toxins, exotoxins and endotoxins. And then what we'll do is we'll just very briefly on Monday hit some main points on the second PowerPoint in medical microbiology, which is epidemiology, reservoirs and transmission, because I'm hoping we can start the last unit on Monday, which is immunology, right? So it's the last unit. Oh my gosh, so fast. Right? And then folks, because I was afraid I would forget to do them, I graded the lab exam 2 study guide packets for the Monday, Wednesday, 8 a.m. lab, so I put them up here, because again, I forget everything, so I thought I better do them now before I forget. Okay, so they're here available for you. Okay, guys, so you'll recall we were working on this first PowerPoint, and I believe we had gotten to the table on comparing and contrasting bacterial endotoxins and exotoxins. Is that right, folks? Were we here? Okay. Good. And we talked about what are toxoids and what are antitoxins, because that will be important vocabulary on lecture exam 3. Okay, so folks, we'll just go through the table here really quickly. And again, this is going to be a short answer question on lecture exam 3 to compare and contrast exotoxins and endotoxins by filling in a table similar to this. So folks, let me just quiz you, okay? So exotoxins are made out of what? What's the composition of exotoxins? They're proteins, yeah? So you guys, as proteins, would they be sensitive to denaturation? Yes, they would be. Good, right? Who can make exotoxins? Which group of bacteria? Gram-positives or gram-negatives, right? All they have to do is have the gene for the exotoxin, yeah, and they can make it. Okay, now in contrast, folks, what are endotoxins made of? Lipid A of lipopolysaccharide found in the outer membrane of gram-negative cell walls. Good, folks. All right. Whew, I know that's a lot, isn't it? So, folks, unfortunately, lipid A is not sensitive to denaturation. We can heat it, you can autoclave it, and it will still remain active, which is bad news for us, right? And, folks, which group of microbes can make endotoxin? Only gram-negatives, right? If it's a component of the gram-negative outer membrane, then that means only gram-negatives can make it. Good job, you guys. All right. The next characteristic, you guys, location. Gesundheit. Location. So with most exotoxins, folks, usually while the bacteria are still alive, they're actively secreting the exotoxin. There's always exceptions, but usually while the bacteria are alive, they're actively secreting the exotoxin. In contrast, folks, with endotoxins, usually we get a lot of endotoxin released only after the bacteria do what? They die. Yeah, they die in lyse. That's when we're going to get massive endotoxin release. And this creates a dilemma, folks, because if you have a patient, and let's say there's gram-negative bacteria growing in their bloodstream or growing in their peritoneal cavity, what's your instinct? What do you want to do to them? You want to kill them, right? Give antibiotics. But you have to be prepared that you could actually trigger massive endotoxin release. And you have to be prepared for the consequences of that, which we'll discuss. Good, you guys. Toxicity, folks, this means, I think of toxicity in like how much toxin is required to cause harm. So the exotoxins are some of the most toxic poisons known, meaning like nanogram amounts can cause serious harm, like the botulinum toxin and tetanus toxin that we're going to describe. They're often said to be some of the most toxic poisons. In contrast, folks, the endotoxin, it takes a lot more endotoxin to bring on harmful effects. But again, the problem is if we have gram-negative bacteria growing someplace in our bodies they shouldn't be, and they start dying, they are going to release massive amount of endotoxin. The signs and symptoms with exotoxins, folks, we're going to see the exotoxins have to bind to specific host cell surface receptors, And for this reason, all the different toxins, they have different targets. They're going to target different cells, different organs. So we can't give a generalized description of signs and symptoms for exotoxins. In contrast, folks, with the endotoxins, we can describe general signs and symptoms. So first of all, they are what we call pyrogenic. And what does pyro mean? Fever producers, right? So usually when you have endotoxin release, you're going to get a fever produced. And actually, did we go over this already, folks? How when the endotoxin is released, it binds to cells of our immune system and causes them to release massive amounts of chemical messengers that we're going to call cytokines. Did we go over this already? Good. Yeah, I didn't want to bore you with that. Good. And as a result of that massive cytokine release, we have a number of changes that can lead to our deaths rather rapidly. So in response to the cytokines, you guys, what's going to happen to our blood vessels? Are they going to get smaller or bigger? Bigger, right? Right? So we have vasodilation. Our vessels will get leaky. We're going to start losing fluid into the extravascular space. And those two events, you guys, what happens as a result of vasodilation and leaky vessels, what happens to blood pressure? It goes down, right? So we call that hypotension. And can hypotension decrease blood flow to organs and cells? Yeah. So we're going to have a decrease in blood flow to organs, right? And as if that's not bad enough, as a result of that cytokine release, we're going to trigger DIC. You remember what DIC was, you guys? Yeah, exactly. Disseminated means it's spread throughout. Intravascular means within the blood vessels. Coagulation. And coagulation, you guys, is just a fancy term for clots. So now we're going to start throwing blood clots everywhere. Now, what will that do to blood flow to your organs? Further decrease it, right? So we have two events that are going on, decreasing blood flow to our organs. And then, you guys, this is just the irony of it all. Because you form so many blood clots, you use up all your clotting factors. So what happens if now you don't have any more clotting factors? You bleed out, right? You hemorrhage. It's just horrific. Yeah, and what happens, you guys, when we start to bleed out, when we start to hemorrhage? What happens to blood flow to the organs? Yeah, is further decrease, right? And you guys, what happens when our organs, our tissues, our cells aren't getting enough blood? They start dying, right? They start shutting down. So we can have what's called multiple organ system failure. That's an old term, you guys, but I do like it. Like your kidneys start to shut down, your liver, your brain, your lungs, your heart starts to shut down. Your patients are going to be confused, right? And that can lead to shock and eventually death. And my understanding is this can happen very rapidly. A patient can progress through the stages of this endotoxic shock, or sepsis, or septic shock. The patient can progress through the stages really rapidly. And my understanding is that what would really help is that if we had a quick and easy blood test to tell when our patients are in the early stages of this endotoxic shock. One of your colleagues said that when she worked in the ER, or they suspected a patient might be in early stages of sepsis, they would take a blood sample and look for lactic acid. And I was like, okay, if I understand this correctly, guys, that makes so much sense, because if your cells aren't getting enough oxygen for aerobic respiration, what can they switch to for short periods of time to make a little bit of ATP if no oxygen is available? Lactic acid, yeah, lactic acid fermentation. So my thought was that's brilliant, because if you have an increase in lactic acid in the blood, that might suggest, right, that the cells aren't getting enough blood, they aren't getting enough oxygen. But right, a good blood test that could detect the early stages of sepsis would probably save a lot of people's lives. With regard, you guys, to the endotoxic shock, septic shock, as we said, it can't kill people, but even in those that recover, if they progress through the late stages, sometimes there can be permanent damage. Like they'll be at higher risk for kidney disease, or maybe have permanent kidney damage. One acquaintance of ours that survived it, neurologically, he was never the same, right? Suggesting there was some kind of brain damage there. So this is a tough one for folks to endure, and even tough when they survive from it. Okay, so we'll just keep going, you guys. So heat sensitivity, if exotoxins are made of proteins, does that mean they should be inactivated by heating them? Yeah, because as proteins, if we heat them, they should denature, unfold, right? And then they should no longer be able to bind to our cells, no longer be able to cause harm. Okay, but we did say, you guys, what's the bad news with endotoxins? They're heat stable. Autoclaving them won't destroy them, right? So I'll make up this scenario, guys. Let's say you have some IV fluids and you think maybe it's contaminated with gram-negative bacteria. And you're like, no problem, we'll just autoclave it and then we can use them. So would autoclaving kill the gram-negative bacteria? It would kill the bacteria, but what's still active and toxic? The endotoxin, right? So if you use that fluid that had those dead gram-negative bacteria in it, the endotoxin is still active, so you could still throw your patient into endotoxic shock. Yeah. Whew. And then, folks, very importantly, can we make toxoids out of exotoxins? Yes, we can, right? That's the basis of a lot of our vaccines, like the diphtheria vaccine, the tetanus vaccine, the acellular pertussis vaccine. Good. Can we make protective antibodies against the toxin? Can we make anatoxin against exotoxin? Yes, we can, right? But now, folks, what about endotoxin? Can we make toxoids out of endotoxin? Nope. Can we make anatoxin, protective antibodies against it? Nope, we can't, right? And that's another thing that would be awesome if we could generate a good endotoxin vaccine. But so far as I know, for humans, they haven't developed a good endotoxin vaccine. Okay. So, folks, let's see here. So what I thought we would do is just take a look at two specific bacterial exotoxins. And then I think the slide after this is just repeating what we said about endotoxins, just to make sure we got all the points. So you guys, the two toxins, the two diseases we're going to talk about is botulism caused by botulinum toxin, and then tetanus caused by tetanus toxin. And you guys, just this morning I screened a couple of pages out of a medical micro book on botulism and tetanus, and I posted them on Canvas under our medical micro module. So it's really good. It's got a lot of good information there. We'll hit some of the main points here, hopefully, in lecture and on the board. Okay, folks. So what's fascinating is the botulinum toxin and tetanus toxin. They're both made by members of the genus Clostridium. And folks, are members of the genus Clostridium, are they strict or obligate anaerobes? Yes, they are. They're strict obligate anaerobes, right? They can't make protective enzymes to protect them against like superoxide anions or hydrogen peroxide. So you guys, again, if it was short answer, are Clostridium, how would you classify them with regard to the relationship to oxygen? Yeah, they're strict or obligate anaerobes, right? They can't live in aerobic environment. But you guys, we're going to talk about that we can get infected stepping on a nail. We could get infected eating dirty green beans, right? So how are the Clostridium surviving in the aerobic environment? What do they make? Endospores, right. So we're going to see you guys often the endospores play a crucial role in pathogenesis and disease transmission. So folks, what I'm going to use here, I'm going to use the these two little babies kind of to trigger me. So we're first going to talk about botulism. And botulism, you guys, is caused by the bacterial pathogen Clostridium botulinum. And botulism is really fascinating, you guys. It's caused by the exotoxin, the botulinum toxin. And what's really fascinating with the disease botulism, we can think of different forms. And two types of botulism, you guys. One is going to be, we're going to classify it as intoxication. What does that mean? right? We're going to ingest preformed toxin. It's not an infection. So we might call this, I don't know, guys, we could just call it generically foodborne, right? So the botulinum toxin will be in some food that we eat. And then the other examples, you guys, are true infections, right? So we're going to be talking about wound botulism. And then the other example is going to be infant botulism. Okay. So folks, with regard to the intoxication, remember this isn't an infection. It's when we're swallowing a poison, a toxin, yeah. And traditionally, this was a problem when we were doing home canning was often a problem. And the high-risk foods are like vegetables that aren't acidic, because Clostridium botulinum, they don't like to grow in acidic environments, right? So the classic one, folks, is like green beans have an alkaline pH, or not an acidic pH. And then in the article, you guys, that I was reading, peppers and mushrooms are kind of like the culprits. And the other one they mentioned in the article, folks, is smoked fish. So just in describing the scenario here, folks, so let's say, well, let me back up here. So folks, we want to recognize that endospores are present in soil, right? And so then, folks, let's take the green beans. Green beans are kind of the classic when it comes to foodborne botulism. So you guys, so you plant your bean seeds, and so where are they growing? You guys are like, what are you talking about? Okay, so you plant your beans, right? They're growing in the dirt, right? So that means the beans themselves could be contaminated with what? What's in the dirt? The endospores, right? So you guys, let's use little balls for endospores, right? So your green beans could become contaminated with endospores. Now the problem is if you pick them and you're going to can them, and the way my mom always did this was in mason jars, right? Well, the problem is you have to make sure that you heat the food sufficiently to destroy what? The endospores, right? And my understanding is it usually requires a pressure cooker, right? Because just boiling won't destroy the endospores, yeah? So if you don't heat the alkaline vegetable, nutrient-vegetable, sufficiently, the endospores won't be destroyed. And in these cans, you guys, this is an anaerobic environment. So what will the endospores do under those anaerobic conditions? They're going to germinate, right? Yeah, the endospores germinate. And that releases the Clostridium botulinum. And what is it going to start making? The botulinum toxin, yeah? So let's say your jar of green beans has been sitting, I don't know, in the garage or basement for a couple of months, and now it's midwinter, right? And you want to maybe make a green salad, and you want to include some green beans. So you open the can, you put the green beans on your salad, and you start eating them. So what are you ingesting? The botulinum toxin, right? Yeah. Now, you guys, a key here is, remember, the botulinum toxin, it's an exotoxin. So would you have prevented this intoxication if you had cooked the green beans really, really well, maybe for quite a while? Yeah, you could have denatured the botulinum toxin, right? But sometimes, like in green salads, right, we don't want to cook the thing, so that would be a problem. So you would ingest the botulinum toxin. And what's fascinating, guys, with the botulinum toxin, what it does is and you guys, in A&P, let me know if I'm making mistakes here, guys. But what the botulinum toxin does, it gets absorbed into the motor neurons. And what it does is it inhibits the release of a chemical messenger, a neurotransmitter, called acetylcholine, specifically at the place where the motor neurons innervate our skeletal muscles. And that's described, you guys, as a neuromuscular junction. And I'll do a horrible cartoon here, folks, but let's pretend this is the motor neuron. Okay, this is the end of the motor neuron. And it's innervating one of our skeletal muscles. So this is a skeletal muscle. All right. So let's pretend, you guys, that's a motor neuron that's innervating my biceps, right? This is my biceps, right? Okay. And let's say I want to bend at my elbow. I need to contract my biceps. So I'm sending a nerve impulse, an action potential. Down my motor neuron. Okay, so here's the signal. Here's a little AP coming down the neuron. And when the action potential reaches the end of the motor neuron, it should trigger the release of this chemical messenger called acetylcholine, right? So when that nerve impulse gets down here at the end, these little vesicles chock full of acetylcholine, I'll use green, okay, are going to get released. And then the acetylcholine is going to bind to receptors on the skeletal muscle cell, and that will trigger all the amazing events that lead to muscle contraction. Okay, so you guys, what does botulinum toxin do? It prevents the release of the acetylcholine. So botulinum, oh my goodness. The botulinum toxin prevents the ACH release. Okay, so it's not getting released. So that means, you guys, can I contract any of my skeletal muscles? No. And so I end up being paralyzed. And it's a type of paralysis, you guys, called flaccid paralysis. Like, I think of it as ragdoll paralysis. I can't contract any of my skeletal muscles. So the result is then what's called flaccid paralysis, like a ragdoll paralysis. And you might think, well, that's not that bad, is it? But what happens is the muscles that we rely on for breathing are paralyzed. So how would we die? We'd suffocate, right? What a horrible way to go, right? So not a good way to go. Okay, so you guys, so that's the foodborne, the intoxication. We're ingesting preformed botulinum toxin, right? So we okay with that one so far? Okay, so what we next want to do, you guys, is take a look at two types of botulism that are caused by actual infections, yeah? And the first one, you guys, wound botulism, this used to be a horrible problem, especially during warfare, battles, before we had antibiotics, right? Because just imagine, you guys, in the old days when they were using swords, to hack at one another, or I don't know, cannonballs to blow people up. If you got injured, what was getting into the wound? Yeah, dirt, right? And what's in the dirt? The endospores, right? So in some cases, folks that were wounded, the botulinum endospores would get in, germinate, start pumping out botulinum toxin, right? And same thing, flaccid paralysis and death by paralysis. Excuse me. But since, we've learned about like antisepsis and now we have antibiotics, that shouldn't happen like in battle settings anymore. But you guys, kind of a heartbreaking thing where you'll see, those of you working in emergency rooms, you might see folks presenting with wound botulism, and very often it's a result that they're IV drug users and they don't have access to clean needles and syringes, right? And so if they're using dirty syringes and needles, right, often they have abscesses. And abscesses, you guys, there's no blood supply there, right? And it might be that, it's hard for them to find like good tissue to inject the drug into. And so what happens if they're literally using, you guys, a dirty needle, right, and they're injecting it into this devitalized tissue, no blood supply, and there's botulinum endospores present, what will happen? They'll germinate, right? And then the people will experience wound botulism. So this is a hazard for the members of our community that are using IV drugs and don't have access to clean needles. In the article, you guys, and I'm not very cool, but they were saying one of the problems is called skin popping, where they're injecting into the skin. They said this is a high risk for wound botulism. So it is an emergency. The good thing is, you guys, they have antitoxin against the botulinum toxin. So that's one way you would treat them. To me, it seems like it'd be really important to find the infected site and clean that up right. So the antitoxin, cleaning up the site, and I would presume antibiotics would also be helpful. In the article here, they said sometimes antibiotics might not be that important, but it seems to me like it would be. You want to kill the Clostridium botulinum. Okay, and then folks, another infection caused by Clostridium botulinum is called infant botulism. And this poor little baby in the slide here, this little baby is suffering from infant botulism, and you can see, there's no muscle tone at all. The baby just totally can't contract any of the muscles, and again, the baby's in danger of dying from respiratory arrest. And probably a lot of you know this already, but you're like, well, how does the baby get infected with Clostridium botulinum? So, folks, it appears that honey, feeding honey to babies, and I think, you guys help me out here, is it you don't want to feed honey to children under 18 months of age? Does that sound right, you guys? Okay, right, okay. And again, you guys, if we think about where does honey come from? So bees are making the honey in their hives, right? But where do the bees gather the nectar and the pollen that they bring back to the hive? What are they visiting? Flowers, right? And you guys, the flowers are growing where? Out in the dirt, right? So flowers, does it make sense, you guys, that flowers could have botulinum endospores in them? And I think the reason the bees are so important is they're concentrating, right? They're potentially concentrating the botulinum endospores in the hive in the honey. But then you're like, wait a minute, this doesn't make sense. How come we can eat honey and we don't get infection, right? So there's two hypotheses, folks, and I think they might be related. Back in the old days, before we knew a lot about the microbiome, I was taught, well, the pH of the baby's intestinal tract when they're really young isn't acidic enough to inhibit the growth of Clostridium botulinum, right? And so remember, Clostridium botulinum, they don't like to grow under acidic pH. And then later is our understanding of the intestinal microbiome developed. People said, well, it might be because the baby's good, beneficial intestinal microbiome isn't fully developed yet. It's not mature enough yet. And it makes sense to me, you guys, well, maybe it's the good members of the intestinal microbiome that are making the acids, right? So that in adults or older kids, the pH within the intestinal tract is acidic enough to inhibit the growth of the Clostridium botulinum. But before that good, healthy, mature intestinal microbiome is established, right? Maybe before 18 months of age, then the babies would be at risk of having germination of Clostridium botulinum endospores in the intestine. So, folks, for reasons I don't totally understand, I know we're an agricultural state and that might be it, but California is supposed to be kind of a hot spot for infant botulism. And indeed, friends of our family, they had a really scary episode where the mom was nursing the baby, and she noticed the baby was having, it looks like, she's like, I couldn't figure out what was going on. The baby was trying to suckle, but the milk was just, drooling out of the mouth. It was like, what's going on? Take the baby to the doctor looks in the mouth and sees this kind of white on the baby's tongue. Oh, the baby has thrush, says the doctor. And so they give the parents like a topical antifungal, I guess, and send them home. But something else is going on, right? So now pretty soon, baby doesn't have any muscle tone, just can't suckle, can't breastfeed, right? So it's like, something's wrong, go back. And you guys, this was so fortunate. They saw another doctor who had just returned from an infectious disease conference where they talked about infant botulism, and he recognized it right off the bat. This is not thrush. This is botulism. So they rushed the baby to the UCD Med Center, and the whole thing is, you guys, is just supporting the baby through the respiratory arrest, right? And eventually they can process the toxin, right? So they got the baby over there in time. The baby survived, and now that baby is this young man. I think he's a history major. He wants to be a teacher, a history teacher. So you'd never know anything happened. But recognizing early, right? Getting the medical support, the respiratory support is crucial, for that. So also, folks, just as we said with the wound botulism, There's antitoxin available, so that little baby received the antitoxin, like the antibodies that would bind to the botulinum and prevent it from binding to the cells of the nervous system. But again, just recognizing early, that's really important. Okay, so folks, that's our botulism. Now, to me, it's really fascinating because the second toxin made by a member of the genus Clostridium is tetanus toxin. So who do you think makes tetanus toxin, folks? Clostridium tetani, right? A close cousin of Clostridium botulinum. And folks, indeed, it's kind of fascinating because when they do the amino acid sequence of the botulinum toxin and compare it to the amino acid sequence of the tetanus exotoxin, they're really similar. And yet, you guys, they act at a totally different part of the nervous system. And the types of paralysis tetanus toxin causes is the exact opposite of botulinum toxin. So it's kind of fascinating. So you guys, so this next exotoxin is going to be made by Clostridium tetani. So the exotoxin, we're just going to call it generically tetanus toxin. And tetanus toxin is responsible for the disease that we call tetanus, or I know growing up, you guys, my mom would call it lockjaw. Lockjaw? And indeed, you guys, the tetanus toxin we're going to discover, it too causes paralysis, but it's the opposite of flaccid paralysis. It's called spastic paralysis. And the reason is all of your voluntary skeletal muscles, they're all contracted. They're all contracted. And the result is the same. The muscles involved in respiration are paralyzed, and so you die from suffocation, right? And again, you guys, I hope I can maybe explain this. So let me see here. With tetanus, you guys, we can talk about, again, it's kind of three forms. So kind of the classic wound tetanus. And then, folks, you could say this next one is just kind of a subset of this, but we'll go ahead and use it, the IV drug. Tetanus, that's just a made-up name, you guys, but just pointing out our at-risk members of our community. And then there is neonatal tetanus. Okay, so if we go through the scenario, you guys, of like the classic wound tetanus, I don't know if your parents told you when you were little not to run around outside in your bare feet. And I know for us it was always you're going to step on a rusty nail and die from lockjaw. That was why we weren't supposed to be running around outdoors without our shoes on. But of course, did we listen? No, we didn't. And then later I found out, oh my gosh, my mom was right. So you guys, this is a foot. This is a foot, I know, right? This is why I don't teach A and B. Okay, and we're running around outside barefoot, yeah? And here's the dirt, and here's the proverbial rusty nail. And you guys, all of this stuff, all parts of the story make sense. A rusty nail has been outside for a long time, getting contaminated with what? Dirt, right? And what's in the dirt? Endospores of? Yeah, so now here again, you guys are going back to those endospores. So here's the Clostridium tetani endospores on the rusty nail. So you step on it, right? And it drives the endospores deep into your tissues. And what's really important, you guys, is you're going to get a lot of damage to your blood vessels. Remember, these endospores, they can only germinate under anaerobic conditions. So the trauma is important. The damage is important. We have to break off blood flow there. So now we have a nice little anaerobic pocket. So what will the Clostridium tetani do, you guys? They're going to germinate and start making what? What are they going to make? The tetanus toxin, right? Okay, so with tetanus toxin, folks, this is so fascinating. It gets absorbed into our neurons, and it travels to the central nervous system, to the spinal cord, I believe. And there, the tetanus toxin prevents the release of what we're just going to call inhibitory neurotransmitters. Okay, so we're going to prevent release of inhibitory neurotransmitters in the central nervous system. We're not peripheral nervous system now, you guys. Now we're central nervous system. And again, you guys, I'll see if I can mangle this description, but this is the best I can understand this. So you guys remember that previous example, and I said, I want to contract my biceps, right? So I start contracting my biceps. Well, as I'm contracting here, I'm stretching the antagonist, is that right, you guys? The triceps, right? And we have these little stretch receptors in our muscles. And the idea is if they get stretched, they're going to contract, right? So for me to contract my biceps, I have to somehow get my triceps to relax, 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 right? And that's the function of these inhibitory neurotransmitters. Yeah, I have inhibitory neurotransmitters being released in my spinal cord so that this antagonistic triceps won't try to contract, right, as it's getting stretched here. Well, if those inhibitory neurotransmitters aren't getting released, folks, that means these paired muscles, agonists and antagonists, there's a battle going on between them. And what will result is whoever is strongest is going to end up being the contracting partner. So you guys, we end up with these horrible postures, like this poor little baby right here, right? Where the muscles are all contracted, and a lot of times in adults we'll end up like our back is bent backwards, like that's extension, right, you guys? Extension, our arms are pulled back, and then we have this terrible expression, you know, as the muscles and the face contract, and you can't open your mouth, right, because the muscles are paralyzed. And again, you guys, so we die from respiratory arrest, right? Not a good way to die. Now, so again, folks, we do have antitoxin available. We have antibodies that can be administered to bind to the toxin to try to prevent, right, more absorption into the central nervous system. Again, support would be respiratory support. That would be the primary thing, right, cleaning out the wound. Yeah. We want to remember, you guys, we have tetanus. We have tetanus vaccinations made of tetanus toxoid. And if we're vaccinated every 10 years, you guys, we should have our own. We should have our own circulating, neutralizing antibodies, antitoxin to protect us from tetanus. That's why if you step on a nail, they right off the bat, they want to see when was your last tetanus vaccine. Our son stepped on a nail just this summer, and I was like, oh my God, we gotta go get a tetanus vaccine. And they looked at his chart and said, no, he's fine. He had a tetanus vaccine just like a few years ago. He's fine. And I was like freaking out. I was like, no, he needs a tetanus. No, they said, no, he's fine. He did not develop tetanus. He's still here, which is good. Okay, so those tetanus vaccines, I think, are really, really important. And often, folks your age, like college students, usually you're behind on your boosters. So if you can, next time you go to the doctor, see if you can check your records and see if you need a tetanus booster, right? Okay, so you guys, so that's kind of like the classic wound tetanus. So the IV drug tetanus, you guys, it is another form of wound tetanus. But again, this is where folks that are using IV drugs and they don't have access to clean needles and syringes, the needle gets dirty and they're injecting again into devitalized tissue, no blood supply, abscess, right, and the tetanus endospores germinate and then they develop tetanus. And then folks, the neonatal tetanus, this is something that's really important for us to know. So this poor little baby right here, you guys, why do you know something's terribly wrong with this poor little baby? What's the first thing you're taught when you hold a baby? You always support the head, right? Because the neck muscles are so weak. What are they doing here, you guys? This is crazy. Yeah. So this is showing that poor little baby is in that spastic paralysis. All the baby's muscles are contracted, right? But luckily, right, we're at some kind of medical facility. The baby's getting respiratory support. So I'm just hoping that baby survived. So how might this happen, folks? So remember when a baby is born, what do we cut when the baby's born? Yeah, the umbilical cord, right? And have you guys seen that little stuff? You know, you're supposed to leave it there until it dries up, you know, and falls off. Well, folks, after you cut it, does the umbilical cord have a blood supply? No. So is it anaerobic? Yes. So folks, if the instruments used to cut the umbilical cord, what if they were literally dirty? What could we inoculate? Tetanus endospores, right? Yeah. And you know, you see in the old West movies, you know, maybe the husband comes in with his hunting knife, you know, and cuts the umbilical cord without sterilizing it, right? It's like, oh my gosh, how did those babies make it, right? And then another thing in the article, you guys, I didn't realize this. Also, if instruments used in circumcision are not sterile, babies can develop neonatal tetanus from contamination of the circumcision site too. And I didn't know about it. I knew about the umbilical cord, but I didn't know that it was an issue with circumcision. So any of those instruments used on the baby, of course, they need to be sterile. Yeah. And an easy way to do it, you guys, is just to heat them, right? If you have some kind of fire, you know, whatever instrument it is, just like you guys do in lab, right? Fire is a great way to sterilize instruments that are going to be used. Yeah. So, okay. And again, folks, we can have antitoxin, antibodies against the tetanus toxin, so that could be administered as well. And I think they say in the article you go ahead and you do give a tetanus booster, right, if the patient is experiencing signs of tetanus. Yeah. Okay. So folks, just is a heads up, and I put this on Canvas as well. Usually on lecture exam 3, I do ask questions on tetanus. And on our final exam, I'm trying to make the final exam a lot of application, like clinical cases or environmental micro cases where you're integrating ideas. So make sure you guys are feeling comfortable with both botulism and tetanus, because you're probably going to see patients present with this either on lecture exam 3 or on the comprehensive final exam. Okay, so you guys, if I describe flaccid paralysis, flaccid paralysis, botulism, ragdoll paralysis, good. And if I describe spastic paralysis, you're thinking tetanus, good. Okay, are endospores important? Yes, right. And if you remember, you guys, Clostridium, it's a stricter obligate anaerobe, right? So if it's going to grow somewhere, it has to be anaerobic. Good. Oh, I have to hurry, don't I? Okay, so you guys, so many of the exotoxins are what we call AB exotoxins, meaning they have 2 functional subunits. And the B subunit, you guys, the B stands for binding. This is a subunit that binds to a very specific host cell surface receptor, right? And it's only when the B binds a host cell surface receptor can the A subunit, the active subunit, get into the cell. Right, and the active subunit, you guys, this is the subunit that's going to cause the mischief, the disruption of normal function within the cell. And so you guys, I was like, oh, this makes sense. So remember we said with botulinum toxin, it targets motor neurons of the peripheral nervous system, and the tetanus toxin targets cells of the central nervous system. So it must be the B, the binding subunit, is binding to different receptors, right? That's why they're going to different places in the body. And furthermore, you guys, an application would be that if you wanted to make a really safe vaccine against an exotoxin, if you could purify, isolate the B subunit, and just use that to vaccinate your patients, what would that trigger production of? Neutralizing antibodies, right? So to me, it seems like that would be a really safe vaccine, right? You don't even have the A component as part of the vaccine. Good, okay. And then, folks, this is just, again, I kind of repeat a lot of this stuff, don't I? But just again, a review on the bacterial endotoxin. They're weakly antigenic. We don't get a protective immune response. We don't get production of neutralizing antibodies. We don't get antitoxin made. We can't make it into toxoids, right? And then again, folks, we were saying the endotoxins, they bind to cells of the immune system, trigger release of that massive amount of cytokines, the cytokine storm. And then we've gone through all of these clinical signs and symptoms of vasodilation, leaky vessels, hypotension, multiple organ system failure, shock, and death. the triggering of the DIC, disseminated intravascular coagulation. Now we have blood clots everywhere. And then eventually, using up the blood clots, we start to hemorrhage out, right? So we can see why we get really reduced blood flow, right? And we also want to remember, you guys, that endotoxin, it's a pyrogen, right? So it's going to trigger what? Fever, right? And you guys, in contrast, what's a pyogen? Pus producer, good. So remember, those two terms look really similar. So, and you guys, that's it, right? So what we'll do, folks, on Monday, we're going to go through the second PowerPoint really quickly. And probably what I'll do is I'll use the medical micro supplemental notes. And the first couple of pages is on reservoirs and transmission. And we're just going to hit a few highlights. So then we can move on to immunology. Yeah, Nia, yeah. No.
Pickling your vegetables prevent.
Well, you see, and this is awesome because I think of pickles as being what? Sour. And sour is acidic. So that should prevent the growth of the Clostridium because they can't grow at acidic pH. That's so important. Like when your Dr. Zenner makes olives, and that's a concern that you might get Clostridium botulinum in there, so he adds some vinegar to lower the pH. So a lot of this stuff, there's really practical application to it. Acidic Clostridium botulinum doesn't do well at acidic pH. Yeah, I love pickles. Anyway, all right. So you guys, talking about pickles, I hope you guys have a good and safe little Thanksgiving break. And just be safe. That's the most important thing. Okay, you guys, take good care. All right. You Yeah, and I'm just gonna close the film really quick, you guys. No. There's that, and also what I keep forgetting, you guys, is until I shut it down, everything people are saying is being recorded, and sometimes people don't want to be recorded. Oh my gosh.